In this set of videos, we're going to go over BLAST, which is one of the most important tools that revolutionized bioinformatics and computational biology, and really biology in general. It's the basic local alignment search tool, hence BLAST. We're going to look at how it works in BV-BRC and how you find things, how it can be used to help you not only look at alignments and see how close things are, but go to disparate sources and find out where those genes are and what they're called in the BV-BRC resource. So the BLAST algorithm takes a query sequence like DNA or RNA or even a protein, and it puts it against a database to find regions of local similarity. BLAST is different than something like Smith-Waterman because BLAST first looked for short regions of high similarity and then would expand out to look at an end-to-end approach. This is very different from what the other algorithms were doing at the time, and it made it very fast. And that's what BLAST is known for its speed. And it's turned out to be pretty accurate as long as you pay attention to the scores, which we'll go over. It helps you find sequences that are similar to the ones you're interested in. So the original publication was in 1990. This publication has tens of thousands, hundreds of thousands of citations. It's a very important publication. And since it originally came out, there are different iterations of BLAST that have been developed. Let's talk a little bit about BLAST scoring. It uses scoring matrices like BLOSSOM 62 for proteins, whereas when it's looking at nucleotides, it's just doing a match-mismatch scheme. It calculates a bit score and an E value, E standing for expectation value. The bit score is just looking at the matches, which get a positive score, and mismatches or gaps, which get a negative score, and it sums those. With the bit score, a higher score means a better alignment. That's in contrast to the E value, which is estimating the number of matches you'd expect to find by chance. In this case, a lower score, a lower E value, means a more significant match. For example, if you had an E value of 0.01, that means there's a 1% chance that the match is random. So you want something that's highly significant. That means it's much, much smaller. There are 4 versions of BLAST that we have in BV-BRC. BLAST-N, which is nucleotide to nucleotide. BLAST-P, which is protein to a protein database. BLAST-X, which is taking your nucleotide query and then searching it against a protein subject sequences or a protein database. And then there's T-BLAST-N, which is taking a protein query sequence and comparing it to a database of nucleotide sequences that are all translated in 6 frames. To show you, for example, if you have a protein sequence, where that protein would be placed on a particular contig in a genome. The job submission interface at BV-BRC looks like this. You choose what the search program you want, you have your query sequence, and you have your database source. So the database source is the most important one, or at least to me, this is the one where BV-BRC really stands out. Many resources offer BLAST. What BV-BRC has that's special and different, perhaps, is it lets you create an individual group of genomes and BLAST against those. And if you have private data, you can BLAST against that specifically if those genomes are in BV-BRC because you've assembled and annotated them or uploaded them. You can do that. You can also search within a taxon and also search within a selected FASTA file. So you can bring your private data in and search against it. And that means you don't have to download BLAST and run it on your computer. You can run it here. So there are advanced options. The default is that there are 10 maximum hits and it has a pretty stringent e-value threshold. So what do the results look like? I always get confused about this, so I printed it out so we can all remember the BLAST terminology. There's a query sequence, which is your sequence, which is your data, what you're bringing in and saying, what is this close to? And then there are the subjects. Those are the sequences in the reference database. And this is what it looks like, your results. It gives you this table, and you just keep in mind, because I put the things that are directly related, the scores directly related to your private data or whatever data it is that you brought in. The query ID is your sequence. The subject ID is the sequence in the reference database that matched your data. That sequence was in a reference genome that you see in that column. In BV-BRC, that sequence has a unique identifier that you see here. Now, if it's something we brought from GenBank or from RefSeq, you will see that it has a locus tag here. Some of them have it and some of them don't because they don't have RefSeq data associated with them. The product is the gene name or the functional name that we give it at BV-BRC, which is generally on the action that that particular protein has, if known, in the organism. The length is the length in nucleotides of the subject within the reference data. It shows you it in nucleotides and also in amino acids. And then the percent identity is saying the percent in the reference that matches your sequence. So now we get to coverage. Coverage is it's taking the whole sequence when I'm comparing either the data I inputted or the reference data, and that's the subject of it. Here it's my data and the query coverage. On my data, in the thing it aligned to, how much of that did it cover? How much of that did it match? What percent of that was matching? And you can have that in the reference too. Pay attention to this, because if you're blasting against a genome, and let's say that genome is in multiple contigs or even one contig, this value will be very low if you're doing a gene or a protein against that, because it's looking across a huge value. Whereas if you're doing blast P and looking protein to protein, it should be better. But this will give you an indication of, is it just hitting part of a gene? Maybe they share a domain, or is it hitting the whole thing? Then we have the score, which we discussed earlier. Higher score means better. And the E value, which lower score means better. So that's how the results are returned. I could have given you a long list, but I think it's easier to see it this way. Then we have the actual alignment. You can click on a little arrow here and it'll open it up. And this is something that you're used to seeing in BLAST at GenBank or other places where you can see where things are similar and where things are different, like here and here. And it gives you the coordinates in the query, your data, versus the subject in the reference. So for the example that we're going to step through in the next videos, we're going to go to this paper because I liked it and because I'm interested in the penicillin binding proteins in bacteria. These are essential proteins that are used when the bacterium is creating its peptidoglycan chain and it's in the process of cell division. We call them penicillin binding proteins, or they were called that historically before we really knew what these proteins are doing. They're essential genes in the bacterium. Penicillin comes in and binds to the proteins, and then the bacterium can no longer divide, so it dies when it does that. And this paper talks about that, and it says that there are 3 proteins, 3 out of the 6 that all Streptococcus pneumoniae have, that are involved in cell division and are the targets of beta-lactam antibiotics like the penicillins. We're going to use BLAST to find out what these proteins are called in BV-BRC and gather other information. Join me in the next videos. The first thing we're going to do is submit a job. So you better watch it so you can see how it's done. See you there. Bye.